Welcome to EPG Patashala. This is Dr. A. Shobana, Department of Biochemistry, Avinashalingam Institute for Home Science and Higher Education for Women. This session we are going to deal about intracellular communications. In multicellular organisms, individual cells become associated in a precise pattern to form tissues, organs and organ systems. Such ordered interactions require that individual cells should be able to recognize, adhere and directly communicate with each other. The ability of the cells to recognize each other was first demonstrated experimentally in 1907 by H.V. Wilson. Since then, cell-cell recognition and adhesion have been explained in a variety of organism cell types. Particularly in animal cells, stable cell-to-cell connections play a key role in organization of cells in tissues. Plant cells also associate with their neighbors not only by interaction between their cell walls but also by the specialized junction between their plasma membranes. So this module is going to deal with the intracellular communication. This module also explains about the cell signaling, types of cell signaling and various receptors involved. After completing this module, you will be able to know about what is cell-to-cell interaction, cell signaling, types of cell signaling and the various cell signaling receptors. First, we will see about cell junctions. Although cell adhesion molecules play an important role in allowing cells to recognize and adhere to one another, the establishment of long-term cell-to-cell connection usually requires specialized structure known as cell junctions. Cell junctions are defined as specialized regions of connections between two cells or between a cell and the extracellular matrix. Animal cells produce three major types of junctions called as tight junctions, plate-bearing junctions, and gap junctions. Each type of junction is designed to perform a particular function like sealing, adhesion, or communication, and plasmodesmata carry out comparable function in plants. Now we will see about what is cell signaling. All cells receive and respond to the signals from their surroundings. The plasma membrane involves in transmitting signals from outside the cell to the cell interior. There is a variety of signaling molecules which are secreted from the signaling cell into the extracellular space by exocytosis. Other molecules are released by diffusion through the plasma membrane and some are exposed to the extracellular space while remaining tightly bound to the signaling cell surface. Signal molecules bind to specific proteins called receptors which are expressed by other cells, thereby integrating and coordinating the functions of many individual cells. In most of the cases, these receptors are transmembrane proteins on the target cell surface. The binding of a signaling molecule to its receptor triggers a change in the receptor conformation. This is in turn initiate a sequence of events that lead to various changes in cell behavior, including metabolism, movement, proliferation, and differentiation. There is a considerable variation in the structure and function of different types of molecules that serve as signal transmitters. Structurally, the signaling molecules range in complexity from simple gases to proteins including small peptides, amino acids, nucleotides, steroids, retinoids, fatty acid derivatives, and dissolved gases such as nitric oxide and carbon monoxide. Some of these molecules carry signal over long distances, whereas the other act locally to convey information between neighboring cells. In addition, the signal molecules differ in mode of action on their target cells. Some signal molecules are able to cross the plasma membrane and bind to the intracellular receptors in the cytoplasm or nucleus, whereas most others bind to the receptors expressed on the target cell surface. Now we will be saying about the modes of cell signaling. The modes of cell signaling are frequently divided into 5 major categories: autocrine signaling, contact dependent signaling, paracrine signaling, synaptic signaling, and endocrine signaling. In autocrine signaling, the cells respond to signal molecules that are produced by them. One important example is the response of the cells to the vertebrate immune system to foreign antigens. Certain types of T lymphocytes respond to antigenic stimulation by synthesizing a growth factor that drives their own proliferation, thereby increasing the number of responsive T lymphocytes and amplifying the immune response. The second one is the contact dependent signaling, where some cells respond to the signal molecules that are expressed on the surface of the adjacent cell rather than being released into the extracellular fluids. This kind of signaling plays a critical role mainly during embryonic development and in immune responses. The third type is the paracrine signaling, where the signal molecules are released into the extracellular spaces and act locally on the neighboring target cells as local mediators. For paracrine signals to be delivered only to the proper target cells, the released molecules must not be allowed to diffuse too far. The fourth type is the synaptic signaling. The neurons or nerve cells typically send long processes that enable them to contact target cells far away. When activated by the signals from the environment or from the other nerve cells, a neuron sends electric impulses rapidly along its axon. When such an impulse reaches the end of the axon, it causes nerve terminals located there to secrete a chemical signal which is called as the neurotransmitter that carries signals between nerve cells at a synapse. The fifth type is the endocrine signaling. Certain signal molecules called as hormones are secreted by the specialized endocrine cells and are carried through the bloodstream to act in the target cell at distant body sites. A classic example is provided by the steroid hormone estrogen, which is produced by the ovary and stimulates development and maintenance of female reproductive system and secondary sex characteristics. In animals, more than 50 different hormones are produced by endocrine glands, including pituitary, thyroid, parathyroid, etc. The endocrine signaling thus controls the behavior of the organism as a whole. The figure shows the different modes of cell signaling. Now, we are going to see what are the classes of signaling molecules. There are hundreds of different molecules that transmit signals between cells. Hence, based on the differences in how far they travel, this chemically diverse group of signaling molecules is divided into three major categories. One is the hormone, second one is the neurotransmitter, and the third one is the local mediator, which we'll be seeing one by one. Hormones are signal molecules which is secreted by the endocrine cells. They are secreted directly into the circulatory system by specialized cells called as endocrine cells and are carried to distant target cells located throughout the body. So based on the difference in chemical structure, hormones can be grouped into three major categories, like steroid hormones, protein and peptide hormones, and amino acid derivatives. Next one is the neurotransmitters. They are released into the synapses from nerve cells and act by binding to the cell surface receptors. The neurotransmitters carry signals between neurons or from neurons to other type of target cells like muscle cells. So there are diverse group of small hydrophilic molecules which is shown in the figure, includes amines like acetylcholine, dopamine, epinephrine, serotonin, histamine, and amino acids like glutamate, glycine, and gamma aminobutyric acid, and peptides like enkephalin and beta endorphins, and the nucleotide ATP. Peptides, enkephalins, and endorphins functions not only as a neurotransmitter at synapses but also as a neurohormones that act on the distant cells. The release of neurotransmitter is signaled by the arrival of an action potential at the terminus of a neuron. The neurotransmitters then diffuse across the synaptic cleft and bind to the transmitter-gated ion channels on the target cell surfaces, where they act to open ligand-gated channels, which is shown in the figure. Many neurotransmitter receptors, including acetylcholine receptor, act as ligand-gated ion channels. Neurotransmitter binding to these receptors induces a conformational change that opens ion channels directly, resulting into net influx of sodium ions in the target cell. This influx causes a membrane depolarization that signals the muscle to contract. The other neurotransmitter receptors are coupled to G protein, a major group of signaling molecules that link cell surface receptor to a variety of intracellular responses. In the case of neurotransmitter receptors, the associated G proteins frequently act indirectly to regulate the ion channel activity, which is shown in the figure. Next one we are going to see is the local mediators. These are nothing but the signal molecules that are produced by a variety of cell types and secreted into the extracellular fluid, where they act on neighboring cells, which is called as the paracrine signaling. In some cases, they also act upon the cells from which they were initially released, which is called as the autocrine signaling. Protein growth factors, which regulate the growth and proliferation of cells in multicellular organisms, are a prominent class of local mediators. Lymphokines are a group of proteins secreted by the lymphocytes. They control cell growth and differentiation. The simple gas nitric oxide is also a local mediator and major paracrine signaling molecule in both animals and plants. So once synthesized, this nitric oxide readily diffuses out of the cell and can act locally to affect the nearby cells. Rather than binding to a receptor on the surface of the target cell, it passes readily across the plasma membrane and alters the activity of the intracellular target enzymes. In mammals, one of its functions is signaling the dilation of blood vessels. The first step in this process is the release of acetylcholine from the terminus of the nerve cell in the walls of the blood vessel. The acetylcholine acts on endothelial cells to make and release nitric oxide that diffuses to the neighboring smooth muscle cells, where it binds to iron in the active site of the enzyme guanylyl cyclase. As a consequence, the enzyme activity is stimulated, resulting into the synthesis of second messenger called cyclic GMP, which induces the muscle cell relaxation and blood vessel dilation. The nitric oxide is also produced as a local mediator by activated macrophages and neutrophils to help them to kill invading microorganisms. In plants, nitric oxide is involved in the defensive responses to injury or infection. Carbon monoxide is another gas that is used as an intracellular signal. Similar to nitric oxide, it acts by stimulating the enzyme guanylyl cyclase. Next, we are going to see about cell surface receptors. Most of the ligands which are responsible for cell-to-cell signaling, that is, which includes neurotransmitters, peptide hormones and growth factors, they bind to the receptors on the surface of their target cells. Consequently, a major challenge in understanding the cell-to-cell signaling is unraveling the mechanism by which cell surface receptors transmit the signals initiated by ligand binding. Some neurotransmitter receptors are ligand-gated ion channels that directly control the ion flux across the plasma membrane. Other cell surface receptors, including the receptor for peptide hormones and growth factors, act instead by regulating the activity of intracellular proteins. These proteins then transmit the signals from receptor to a series of additional intracellular targets, frequently including transcription factors. Ligand binding to a receptor on the surface of the cell thus initiates a chain of intracellular reactions, ultimately reaching the target cell nucleus and resulting in the programmed changes in gene expression. So based on the mechanism of action, the cell surface receptors has been divided into three major classes. One is the ion channel linked receptors, second one is the G protein linked receptors, and the third one is the enzyme linked receptors. First, we will see about ion channel linked receptors. They are nothing but the transmembrane proteins that function as ion channels and are referred to as ion channel linked receptors. Also known as transmitter gated ion channels or ionotrophic receptors, are involved in a rapid synaptic signaling between electrically excitable cells. This type of signaling is mediated by a small number of neurotransmitters that transiently open or close on the ion channel formed by the protein to which they bind. This kind of receptors belong to a large family of multipass transmembrane proteins. A good example is acetylcholine that triggers the contraction of the muscle cells, which slows the heartbeat and transmits signals from cell to cell. Acetylcholine receptors reside in the plasma membrane of vertebrate skeletal muscle cells and the electric organs of certain species of fish such as electric ray. The figure illustrates a regulation of permeability of ion channel by acetylcholine. The binding of acetylcholine to the receptor's alpha subunit induces a conformational change that causes the channel to open, allowing the sodium ion to diffuse through the channel. The open configuration of the channel is unstable and is superseded by a closed conformation whose energy is very lower. The acetylcholine molecules then dissociate from the receptor and are hydrolyzed by the enzyme acetylcholine esterase. After the release of the bound acetylcholine, the receptor returned to its original configuration. The next one we are going to see about is the G protein linked receptors, which plays an important role in signal transduction. G protein linked receptors form the largest and the most diverse group of cell surface receptor and is found in all eukaryotes. More than thousands of such G protein linked receptors have been identified in mammals, including the receptors for many neurotransmitters, neuropeptides and peptide hormones. The G protein linked receptors are structurally and functionally related proteins characterized by 7 passed transmembrane proteins, which is shown in the figure. The binding of the ligands to the extracellular domain of these receptors induces a conformational change that allows the cytosolic domain of the receptor to bind a G protein associated with the inner phase of the plasma membrane. This interaction activates the G protein, which then dissociates from the receptor and carries the signal to an intracellular target, which may be either an enzyme or an ion channel. The G proteins are large heterotrimeric GTP binding proteins. They are constructed from 3 subunits designated as alpha, beta and gamma, whose interactions are very crucial to the regulation of the enzyme adenylyl cyclase. In the absence of a stimulating hormone, GDP is bound to alpha subunit of the G protein, and the alpha, beta and gamma subunit remain associated with one another. So in this configuration, the G protein cannot activate adenylyl cyclase. Hormone binding induces a conformational change in the receptor such that the cytosolic domain of the receptor interacts with the G protein and stimulates the release of bound GDP and its exchange for GTP. The activated GTP bound alpha subunit then dissociates from the beta and gamma and stimulates the enzyme adenylyl cyclase. The activity of the alpha subunit is terminated by hydrolysis of bound GTP, and the inactive alpha subunit, which is now bound with the GDP, that reassociates with the beta gamma complex and is ready for the cycle to start again. About 20 different alpha subunits, 4 beta subunits and 7 gamma subunits have been described in mammalian genomes. Different G proteins associate with different receptors. So this array of G protein coupled receptors to distinct intracellular targets. The G protein associated with the epinephrine receptor is called as the stimulatory G protein, GS, because its alpha subunit stimulates the enzyme adenylyl cyclase, causing an increase in the cyclic AMP formation and resulting an increase in the heart rate and contractile force. The other G protein subunits alpha and beta gamma act instead to inhibit the enzyme adenylyl cyclase, causing a decrease in the cyclic AMP formation. The third one we are going to see is the enzyme linked receptors. They act as a receptor for signaling a ligand or directly as enzyme. So this is the third group of cell surface receptors in which the receptors are directly linked to intracellular enzymes. Like G protein linked receptors, enzyme linked receptors are transmembrane proteins with their ligand binding domain on the outer surface of the plasma membrane. The cytosolic domain either has an intrinsic enzyme activity or associate directly with the enzyme. The enzyme linked receptors are formed from a single pass transmembrane proteins and are heterogeneous in structure, which is shown in the figure, as compared with the other 2 classes. There are 6 classes of enzyme linked receptors which have been so far recognized. One is the receptor tyrosine kinases. Second one, tyrosine kinase associated receptors. The third one is receptor like tyrosine phosphatases. Fourth, receptor serine or threonine kinases. Fifth, receptor guanylyl cyclases. And sixth one is the histidine kinase associated receptors. Among all these, the largest class of enzyme linked receptor is the tyrosine kinases and serine or threonine kinases, which constitute the second major class of the enzyme linked receptor. So we are going to see about tyrosine kinase receptors. They catalyze the phosphorylation of amino acid tyrosine when a signaling ligand binds to the receptor exhibiting a protein tyrosine kinase activity. The receptor phosphorylates itself on several tyrosine residues. The tyrosine phosphorylated by the process of autophosphorylation are located in the receptors of cytoplasmic domain, that is, the region of the receptor exposed at the cytoplasmic surface of the plasma membrane. The phosphorylation of tyrosine serve as selective binding site that interacts with specific cytoplasmic molecules, activating a cascade of events that triggers the appropriate cellular responses to the signaling growth factors. By now, more than 50 receptor protein tyrosine kinases grouped into 16 structural classes have been identified, including the receptors for epidermal growth factor EGF, platelet-derived growth factor PDGF, fibroblast growth factor FGF, hepatocyte growth factor HGF, vascular endothelial growth factor VEGF, macrophage colony stimulating factor MCSF, ephrin, insulin, and all the neutrophils, including nerve growth factor NGF. All these receptor tyrosine kinases share a common structural organization: a N-terminal extracellular ligand binding domain, a single transmembrane alpha helix, and a cytosolic C-terminal domain with protein tyrosine kinase activity. So most of the receptors protein tyrosine kinases consist of single polypeptides, although the insulin receptor and some related receptors are dimers consisting of two pairs of polypeptide chains. In some receptors, tyrosine kinase domain is interrupted by a kinase insert region, example VEGF receptor. Several families of receptor tyrosine kinases that operate in mammals are given in the figure. The binding of ligands, example growth factors, to the extracellular domains of these receptors activates the cytosolic kinase domains, resulting in the phosphorylation of both the receptor themselves and the intracellular target proteins that propagate the signal initiated by the growth factor binding. The first step in signaling from most receptor protein tyrosine kinases is the ligand-induced receptor dimerization. Some growth factors, for example PDGF, is a dimer which cross-link two receptors together. Other growth factors such as EGFs are monomers but binding to two distinct receptors simultaneously cross-link them, inducing conformational changes that favor the receptor dimerization. Ligand-induced dimerization then leads to the autophosphorylation of the receptor as a dimerized polypeptide chain cross-phosphorylate one another, which is shown in the figure. The receptor for insulin acts slightly in a different way. It is a tetramer to start with, and ligand binding is thought to induce a rearrangement of the transmembrane receptor chains so that the two kinase domain come close together. Now, we are going to see about the pathways of intracellular signal transduction. Most cell surface receptors stimulate intracellular target enzyme, which may be either directly linked or indirectly coupled to receptors by G proteins. These intracellular enzymes serve as a downstream signaling elements that propagate and amplify the signals initiated by the ligand binding. So in most of the cases, a chain of reaction transmits signals from cell surface to variety of intracellular targets, a process called as intracellular signal transduction. The targets of such signaling pathways frequently include transcription factors that function to regulate gene expression. Intracellular signaling pathway thus connect the cell surface to the nucleus, leading to changes in the gene expression in response to extracellular stimuli. Now we are going to see about a second messenger called as cyclic AMP. The intracellular signaling was first elucidated by studies of the action of hormones such as epinephrine, which signals a breakdown of glycogen to glucose in anticipation of the muscular activity. In 1958, Earl Sutherland, a scientist, discovered that the action of epinephrine was mediated by an increase in the intracellular concentration of cyclic AMP, which leads to the concept that cyclic AMP is a second messenger in hormone signaling, the first messenger being the hormone itself, which is the extracellular signal. Cyclic AMP is formed by the ATP by the action of plasma membrane bound adenylyl cyclase and degraded to AMP by cyclic AMP phosphodiesterase. The normal concentration of cyclic AMP inside the cell is about 10⁻⁷ molar, but an extracellular signal can cause this cyclic AMP levels to change by more than 20 folds in seconds. The epinephrine receptor is coupled to adenylyl cyclase via a G protein that stimulates enzymatic activity, thereby increasing the intracellular concentration of cyclic AMP. The effects of cyclic AMP in animal cells are mediated by the action of cyclic AMP dependent protein kinase, which is also called as protein kinase A, which is an enzyme discovered by the scientists Donald Walsh and Edwin Krebs in the year 1968. Its widespread occurrence in eukaryotic cells and its ability to catalyze the phosphorylation of a variety of proteins has led to the idea that most other biological effects of cyclic AMP are mediated by protein phosphorylation. In the regulation of glycogen metabolism, protein kinase A phosphorylates two key target enzymes. One is the phosphorylase kinase and another one is the glycogen synthase. The elevation of cyclic AMP and activation of protein kinase A thus blocks further glycogen synthesis at the same time as it stimulates the glycogen breakdown. In many animal cells, increases in cyclic AMP activates the transcription of specific target genes that contain a regulatory sequence called as cyclic AMP response elements CRE. In this case, the signal is carried from the cytoplasm to the nucleus by the catalytic subunit of protein kinase A, which is able to enter the nucleus following its release from the regulatory subunit. Within the nucleus, this protein kinase A phosphorylates a transcription factor called as CREB for CRE binding protein, which leads to the activation of cyclic AMP inducible genes. Although most effects of cyclic AMP are mediated by protein kinase A, the cyclic AMP can also directly regulate ion channels, which is independent of protein phosphorylation. Cyclic AMP functions in this way as a second messenger involved in sensing smells. Many of the odorant receptors in sensory neurons in the nose are G protein coupled receptors that stimulate the adenylyl cyclase, leading to an increase in intracellular cyclic AMP. Rather than stimulating protein kinase A, cyclic AMP in this system directly opens the sodium ion channels in the plasma membrane, which leads to the membrane depolarization and initiation of nerve impulse. The next second messenger what we are going to see is the cyclic GMP, which is also an important second messenger in animal cells. Cyclic GMP is formed from GTP by guanylyl cyclase and degraded to GMP by phosphodiesterase. There are different types of guanylyl cyclases which are activated by both nitric oxide and the peptide ligands. Stimulation of these guanylyl cyclases leads to increase in the cyclic GMP levels, which then mediate biological responses such as blood vessel dilation. The best characterized role of cyclic GMP is in the vertebrate eyes, where it serves as a second messenger responsible for converting the visual signals received as a light to nerve impulses. The photosensitive molecules in rod cells of the retina are G protein coupled receptors, which is called as the rhodopsins. The plasma membrane surrounding the outer segment contains cyclic GMP gated sodium ion channels. These channels are kept open in the dark by cyclic GMP that are bound to them. Rhodopsin is activated as a result of absorption of photon of light by a covalently attached chromatophore 11-cis retinal, which then isomerases to all trans-retinal, inducing a conformational change in the rhodopsin protein. Rhodopsin then activates the G protein transduction GT, causing its alpha subunit to dissociate and activate cyclic GMP phosphodiesterase. The phosphodiesterase then hydrolyzes cyclic GMP, leading to decrease in the intracellular level of the cyclic GMP. So this drop in the cyclic GMP concentration lead to a decrease in the amount of cyclic GMP bound to the plasma membrane sodium ion channels, allowing more these highly cyclic GMP sensitive channels to close. So in this way, the signal quickly passes from the disc membrane to the plasma membrane and a light signal is converted to electric one. The closure of this sodium ion channels in the plasma membrane hyperpolarizes the membrane and consequently reduces the rate of neurotransmitter release from the synaptic region. Because neurotransmitter acts to inhibit many of the postsynaptic retinal neurons, illumination serves to free the neurons from inhibition and thus in effect excites them. Now we'll just see what is phosphoenocetic signaling pathway, which is an alternative second messenger system, which involves the use of second messenger derived from the inositol phospholipid, which is called as phosphatidyl inositol 4,5-diphosphate, PIP2. It is a minor component of plasma membrane and is present in the inner leaflet of the phospholipid bilayer. The next one is MAP, mitogen activated protein kinase pathway, which controls a variety of cellular responses. So this pathway refers to a cascade of protein kinases that are highly conserved in evolution and play central roles in signal transduction in all eukaryotic cells ranging from yeast to humans. The central elements in the pathway are a family of protein serine or threonine kinase, which is called as the MAP kinases, that are activated in response to a variety of growth factor and other signaling molecules. Next one is the Jackstart signaling pathway, which provides a fast track to the nucleus. MAP kinase pathway provides indirect connection between the cell surface and the nucleus, in which a cascade of protein kinases ultimately leads to the phosphorylation of transcription factor. The Jackstart, which is called as the Janus kinases, signal transducers and activators of transcription signaling pathway, however, provides a much more immediate direct connection between protein tyrosine kinases and transcription factors. To conclude this session, from this module, we have seen what is cell junctions, and we had a clear picture about cell signaling and the different modes of cell-cell signaling like autocrine, contact dependent, paracrine, synaptic and endocrine signaling. We have also seen the three classes of signaling molecules like hormones, neurotransmitters and local mediators. We also came through three major classes of cell surface receptors, that is ion channel linked receptor, G protein receptors and enzyme linked receptor, which is the tyrosine kinase receptors, and a detailed picture about these receptors also we have seen. So this module was also dealt with the pathways of intracellular transduction, cyclic AMP and cyclic GMP as a second messenger system. Apart from this, the alternative to these second messenger, that is phosphoenocetic signaling pathway, MAP kinase pathway and Jackstart kinase pathway were also seen. Finally, we conclude that intracellular signaling pathways thus connect the cell surface to the nucleus, which leads to changes in the gene expression in response to extracellular stimuli. Thank you.